Well, thank you all for coming. As everybody gets settled and grab some lunch, I'm really pleased to be able to present this seminar series to you once again with the support of the CCTS. We always like providing the space and of course the lunches. And it really is a nice example of translational science, I think, that the whole drug discovery and development process. So we'll be meeting every Friday for about 10 weeks. We have 1 week off, and hopefully you've all seen the schedule. And we'll kind of go through the drug discovery and development pipeline. And we'll spend an hour today as an overview, introduction seminar, and I'll refer to different seminars that will go into different topics in depth as we go through the slides. And then in the coming weeks, if you have a specific interest, let's say you want to learn more about medicinal chemistry, but you really know already everything there is about high-throughput screening, you can just kind of pick and choose which seminars you want to attend. And we all have local speakers that are experts in their particular topic, both from Southern Research and from UAB. So I hope to see many of you in the coming weeks ahead. So today I will give you kind of an overview and set the stage of what the drug discovery and development landscape looks like, and also how it's paid for. As you probably have heard, drug discovery is an expensive exercise. Sometimes you hear numbers of between $1 and $2 billion. That's what it takes to make a drug. So there's some truth to that, but I'll go in a little bit more in depth to show you where those numbers come from. And I also want to talk to you about the Alabama Drug Discovery Alliance. So the CCTS, as you may know, has a drug discovery component to it, and that's kind of folded into this alliance that we have here locally between UAB and Southern Research. So together, we think we can hit all those points that I'll talk to you about today, what's needed in drug discovery and development. So it was only logical that we would join hands and enter this alliance together. So my name is Maaike Everts. I'm the director of the ADDA, and if you ever forget how to spell my name, if you need to send me an email, you can also use adda@uab.edu or Alabama Drug Discovery. It's easier to remember than how many vowels are in my name. Just acknowledge that. So I will talk to you first about the drug discovery and development pipeline, then we'll go through the different topics and parts of that. Then I'll talk a little bit about what kind of funding opportunities are available at NIH and also some foundations. And then, like I alluded to, what we can do locally with the ADDA. So let's start with the pipeline. So this is the type of picture you see many times in drug discovery and development lectures, where on the left hand of the slide, you start with really basic biology. Here it's identified as target identification, and I'll talk to you about what a target is. And then here's this cutout, blown up part that talks about target identification, hit generation, and lead generation. And this is really a very early stage in the drug discovery and development process, even though this can already take many years. But this is really still early stage research and discovery, finding a molecule that maybe one day may become a drug. Then from that leads into typically preclinical studies in animal models, so mouse models, maybe whatever animal is suitable for your particular disease, to show the concept that this potential drug can treat your disease of interest. And also you're going to look at toxicology and things like that, and we have a special session on that topic. And that then sets the stage to go into clinical trials. And that's a pretty long phase. It actually consists of 3 different parts. Phase 1 is typically a really small study where you really look at safety, because this is the first time a drug touches a human being. Then in phase 2, you really look at, does the drug actually treat the disease, yes or no, in a control population and a therapeutic population. And then finally, phase 3 trials, those are typically really big trials done at multiple sites across the country. They cost a lot of money. You enroll thousands of patients, and executing those trials takes several years. But ultimately, then you go to the FDA, and the FDA needs to review all the information from those trials and then hopefully approves your drug. And then finally, you can start selling your drug. So this process takes typically 12 to 15 years. So this is definitely not an instant gratification kind of process. So what we do here locally is really mostly focus on this first piece here at the top. And what you can see, we can probably get something to maybe a point where it's ready to go into clinical trials. But then there's still a lot that needs to be done before you can ever get it to sell it to a patient. So this is kind of the outline, the pathway that we'll follow today. We'll start with target identification, validation. We'll talk about assay development. I'll talk about what that means and why we need to do it when we start to screen with robots. And we start to do screens, a primary screen, which is a big funnel where we screen through thousands and thousands of molecules and see if there's anything that works like we want it to. Something called secondary screens, I'll explain that later. Then you start into a chemistry phase called lead optimization, preclinical development, and then the clinical development I just talked about, ultimately hopefully leading to regulatory approval. And I'll just kind of have this slide pop up a few times to show you where we are in this process as I'm talking you through it. So let's start with the beginning, a target identification and validation. And first, let's talk about what a target is. What's the definition of that? So it's a naturally existing cellular or molecular structure that's involved in a disease that a drug in development is meant to act on. So you really need to understand on a molecular level what the mechanism is why disease occurs. What proteins are not talking to each other, or maybe they talk to each other too much, or what piece of DNA is being transcribed too much. Things like that. You really need to understand that as granularly as possible. And even if you know maybe a pathway that is, let's say, overactivated or inhibited, there's usually different components in the pathway, all these molecules that talk to each other in a cell. Well, which molecule is the best one to really find a drug for that can inhibit its action to have the effect? Because sometimes you can end up with a whack-a-mole effect where you inhibit expression or activity of one protein and then another one kind of takes over and does its function. So the more you understand about this basic biology, the better you're off to defining if a target is a good idea or not. So you really need to understand that this really comes out of basic research. And of course, we do a lot of this at UAB. So that was also one of the a reason to start the ADDA. We have all this basic research here on campus. We really need to do something with it beyond just talking about how this might be a good target for future drug discovery. Let's actually do it. But how do you know it's a good idea? Of course, you have the hypothesis in your biology and understanding how proteins talk to each other in a cell. But if you maybe at least have some evaluation in a cell culture system, for example, using, let's say, siRNA or something like that to knock out the function of the protein, do you see a difference in how the cell behaves, yes or no? Very easy examples, maybe cancer. If you have a cancer cell that's dividing normal at a really high rate, if you knock out this particular protein that you think is a good target, does it stop dividing? You know, those kind of easy questions you can answer. Then if that looks somewhat good, you can go into mouse models or rodents, rats maybe, whatever is suitable for your disease. If you have knockout mice, maybe CRISPR-Cas or whatnot, can you recapitulate the phenotype of this mouse? Does this mouse have asthma or whatever your particular disease of interest is? Is there an animal model that kind of gives you some confidence that your target is an important one? What's really helpful is if you have clinical data. Either protein or gene expression levels and maybe disease tissue versus healthy tissue, things like that. Correlation, of course, is not causation, but it does give you a bit of a leg up. If there's a difference in activity of your target in healthy versus sick people, it may give you an idea that this is an important target to go after. But unfortunately, we really don't know if a target is valid or not until we have a drug that works in humans. So many times, all the data in cell culture systems and animal models point to this is really important, but when you do ultimately the clinical trial, you see no effect. I think a good example is the Alzheimer's field. We've had so many phase 3 clinical trials going on. Animal models seem to support that, going after these particular targets is a good idea. When you test it in humans, it doesn't work. So this is why sometimes companies make what we call Me Too drugs. So if one company finds out that this target is a good idea and they have a drug that's in development and shows efficacy, then maybe another company will go in like, okay, they took the risk and they showed that targeting this is a good idea to help disease. Now it's de-risked, so maybe now we want to invest our money in to kind of do the same thing. So that's called a Me Too drug. So they may have a different molecule, but maybe the target is the same. So there's a lot of criteria that go into evaluating whether a target is a good idea or not. So I listed some over here. I'm not going to go through them in detail. But what we look for here locally is it novel? Is there maybe some new biology that's been discovered that really nobody else in the world knows? And the UAB or some research investigator is the actual expert on this. And it hasn't been published yet. Do we have a leg up on the competition? So that's a good idea to invest our resources into discovering a drug for this. And something called druggability plays a role. And there's really not a good definition for it, but it mostly means if the target is in a class of targets that other drugs exist for. So, for example, kinases. They're a huge class of targets, particularly in oncology. We know we can develop a small molecule that inhibits a kinase. There's hundreds of them, but they all function somewhat the same. So we know that in principle, if we discover a new kinase, there's a pretty good chance we can find a small molecule that can inhibit it. Now, in contrast, people up until fairly recently, and still people sometimes think this way, is when you want to disrupt protein-protein interactions. So proteins, when they bind to each other, usually they have a large surface area with lots of hydrophobic interactions, so they really stick together. Can you actually design a small molecule that can worm its way in between those two proteins and disrupt it, yes or no? That's a much more difficult question to answer than, let's say, develop a new kinase inhibitor. So those targets are considered a little bit more risky. There are examples now where we actually, within our pipeline, where we've been able to find molecules that do this, but it's still considered less druggable than some other things. Also, you want to look at selectivity of targets, especially when you think about tumors. If you inhibit the expression of a certain protein or activity of it, if it would be expressed everywhere in your body and it has a critical function for the function of your body, you probably don't want to block it because it's going to cause a lot of side effects. So things like that. And then the last point on this slide is intellectual property potential. And in an academic environment, we're sometimes not really primed to think about this, but in drug discovery we have to, because it is such an expensive process to go through. It may not take 1 billion if you were to make good choices from the get-go, but still you're talking about hundreds of millions of dollars. UAB doesn't have that laying around, right? Even though we have a capital campaign, probably not the 500 billion or 1 billion that we're raising, probably shouldn't go into one project. So at some point, you need to collaborate with an external entity who has lots of money. So pharmaceutical company, maybe a biotech, venture capital firm, things like that. They ultimately want to make their money back because they're for-profit institutions. That's their business model. In order to make your money back, you need to have intellectual property around your molecule because that allows you to block other people from selling your drug. So it's absolutely critical to do this because if this doesn't exist, then you're never going to find an investor in your program and your potential drug will never see a patient. That's just a reality that we live in. So we always look, especially when it comes to composition of matter intellectual property, like is there a space where we can operate? Can we plant a flag around a certain shape of a molecule that nobody else has claimed yet? So we look at that. So we have a lecture on target evaluation and identification on March 30th to go a little bit more in depth about what makes a good target, yes or no. But once you've identified, okay, I want to go after this target, I went into PubMed and or in my research, I found out that this enzyme is absolutely critical for development of disease. So now what you do? Well, one of the ways you can go about finding a new molecule is with high-throughput screening. So it's kind of a lots of shots on goal approach. So here's the definition of high-throughput screening. It's a system that uses specialized automation equipment, or robots, and high-density microtiter plates to screen a large number of wells in a short period of time. So we can easily screen 30,000 to 100,000 compounds per day once you've set it up. So here's a picture of the robot at Southern Research. It kind of has a wrist and an arm and an elbow, and it kind of can rotate around itself to go to different stations. It can move along a beam to manipulate those microtiter plates that are about this big and have things done to that plate. So here you can see, a stack of these plates that are being processed with the little barcode on it. So it's kind of critical that you set up a lot of things before you can hit go on the robot system. So for example, you need to really manage all your compounds that you are going to be screening. So Southern Research is close to 1 million compounds in your collection. We typically screen 200, 300,000 in our projects. So you need to remember which compound came from which vendor, in which well is it, in which freezer, how many times did the plate get frozen and thawed, all those kind of things. So you need to have specialized software that takes care of that. Of course, you need to have...
You said Southern Research is 1 million library that covers all possible
chemical space?
Yeah, chemical that you target between whatever protein or it's 1 million is just common right now.
So the size of the library depends a little bit where you are. What's good or not. So I think typical pharmaceutical companies look at about 1 million or so. But in academic institutions, they usually have a much smaller library. So you actually, even with the 1 million compounds, you really cannot cover all chemical space because it's at infinity. It's like 10 to the 15th or something possible permutations. But what you want to have in your collection is a broad variety, but then also have molecules that are somewhat drug-like. So they're not, 10,000 Daltons with 50 hydroxyl groups and things like that that chemists will look at and start laughing. So some of these compound collections you can purchase, and they curate those collections and they offer them, just for sale on the internet. And they may be tailored to these compounds should be more prone to inhibitor kinase, or these should be more prone to get into blood through the blood-brain barrier, things like that. Or maybe they're a collection of natural products that have shown some activity in the past. But somehow, though, you really need to think of high-throughput screening. Somebody compared it once that instead of finding the needle in the haystack, it's actually getting rid of a lot of hay. So what you really kind of do is screen out all the compounds that don't have any effect at all. And what you then do is use the molecules that are left as a starting point to do medicinal chemistry. So there is hopefully somewhat high likelihood you're going to find something in your compound collection, but it's not guaranteed, and you don't know if it's actually going to eventually work out to become a drug because you need to do a lot of optimization. But a lot of companies do tend to have similar type libraries. So sometimes you can actually come up with molecules that even at two completely different institutions, let's say here in Birmingham and let's say Eli Lilly does a similar screen, we may end up with somewhat the same molecules because it's just the way these compound collections are put together tends to be done by the same type of chemists who have the same training and things like that. So because you have so many compounds, you also need to have informatics associated with this, again, to help identify what's the data that's associated with one compound. And actually at Southern Research, they have software, they call it Dotmatics, where it's kind of an Oracle-based software where they can pull up a compound and associate all the data that was ever generated for this compound, because the same collection gets screened in multiple different screens. So there may be data points on antiviral screens, oncology screens, protein-protein interactions, and so you need to have it all in one place to make the most of your information. And you need people that know how to do this. Of course, at some point the robot executes, but setting up the robot and troubleshooting, you need experienced people to do that. And that was a really nice advantage of having Southern Research here locally in Birmingham to start the ADDA. They had that all in place. So around the same time, there were a couple of other academic drug discovery centers that were started around the country. I go to those meetings and you hear the directors talk about, we started with a $100 million anonymous gift. And you think, that would be nice. We didn't have that. But we had Southern Research where they already had the infrastructure in place. So those other institutions needed to hire ex-pharma people, buy robots, set up their collections, all that kind of stuff. We had that right across the street already. So we were rocking and rolling before anybody else even got their equipment set up. So Bob Bostwick is the director of high-throughput screening at Southern Research, and he will give a lecture next week on March 23rd on high-throughput screens. And he has videos and things like that.
Sure. I have a general question. Like you said, Southern Research has 1 million compounds. So are these unique? Or so did Southern Research synthesize, design all these compounds, or just purchase from other commercial?
Yeah. So the 1,000 compound collection consists mostly from commercial vendors, but there's a subset of about, I think, 20,000, 25,000 compounds that have just been generated throughout the history of Southern Research. So they exist this year for 76 years. So they have a lot of compounds in a collection that nobody else in the world has.
So the related questions, if I say, screen at Southern Research, do not give, expected outcomes. So is it reasonable or possible to do this assay in a different place, a commercial company or NIH sponsored screening center, maybe
find something
using
different.
different libraries?
Yeah.
Okay.
It depends a little bit. So in some screens that we did, especially in the first few years of the ADDA, we had limited. We only screened sometimes 80,000 compounds. because we were limited by the amount of protein that could be produced that could be used in the assay. So 80,000, you are starting to limit yourself a little bit of what chemical space that was covered. So for example, that particular project is Alzheimer's project. We do propose to screen more because we do think we haven't touched upon a lot of types of libraries that have unique structures because we just had to pick and choose what would go in that 80,000. So in that case, it does make sense. The 200, 300,000 that we now typically screen also includes FDA approved and unapproved drugs. So we always immediately have a repurposing screen kind of built in. If you have really, maybe your assay is just not set up in order to find the answer that you're looking for. And that, no matter how many libraries you're going to screen, you're always going to get subpar answers. And that's why assay developing the assay in a way that it gives you useful information is so critical. And it's a more difficult process than most people realize. So yeah, I think it depends on how many compounds you screened initially and how reflective that is of the library as a whole. So the chemist can kind of opine on that. And the Southern Research has the MLPCN library that's about 300,000. So they were a site for NIH to do their screening. When you go to NIH and you apply for a grant, that they do the screening for you, like you have at R21. Mm-hmm. That was outsourced to different centers around the country, and Southern Research was one of those centers. And they all had the same library at all those separate places. So this is a picture of the microtiter plates that are being used. So here on the left, you see a 96 well plate that most of you are maybe familiar with 8 columns and 12 rows. But we typically use 384 well plates or 1536 well plates because you have to use a lot less volume to run your reactions. And then we also have 3,456 well plates, really small volume, but that's typically you start to run into trouble just designing the assay so you get meaningful information because your wells are so tiny that you start to get issues with your compounds sticking to the plastic, things like that. So these two middle ones are the sizes we typically run in, and it just depends on the assay performance, which type we do it in. But you want to really use robots to do your pipetting for you at this stage, because if you need to go like, okay, 20 over, 41 down, you're going to go kind of crazy. And they have really cool robots, and I think Rob will have a picture of one next week that use acoustic waves to shoot droplets into different wells in a very controlled manner. So yeah, robots are much better at pipetting than humans. Just so you know. So how we generally approach things is that we develop an assay that can be done by the robot, and then we first do a screen of 10,000 compounds and duplicates in 2 different days, and we see, do we get the same results, yes or no? And then typically, like I said, we screen between 100 and 300,000 compounds when we hit the go button. And then about 2,000 or so hopefully show up as active. But this is only done at an N of 1, at 1 concentration. So what you want to do then is what we call cherry picking, is you grab the active molecules from all different plates and put them on a new one, and then we test those at 10 different concentrations in dose response. And the idea is if you have high concentrations of the compound, you should see a large effect. When there's almost nothing in there, you should see a low effect, and you can plot curves through those data points and determine what we call the IC50. And that shows, how good is this compound at doing what we want it to, and you can rank order them, things like that. At this point, many times we do a secondary or counter screen. So with the kinase example, this is a good one. There's a lot of kinases out there, but you really may want to inhibit just one and not another. Where at this point, you can do in your first screen, you screen for your kinase of interest, but in your dose response, you may do both of them side by side, so you immediately have that data, how well does my compound inhibit the kinase I want versus the one I don't, and you can, again, compare and contrast those. Maybe you also want to run a toxicity screen at that point in time if your compound is not supposed to kill a cell, to, again, give the chemist information to select which ones look the most attractive. Because at that point, you generate a hit list, and the chemists get into a room and they start to, with the big red pen, start going, nope, nope, maybe, nope, nope. They're a cranky bunch. They will admit that themselves. But they will really look at it from a structure perspective. Is this something that we can start playing with and modifying? Are there any structural alerts that we need to be aware of? And Corinna Urgeli-Zafran, she's the director of chemistry at Southern Research. She's going to give a talk about that in particular. So then it depends a little bit on the number of hits, what happens next. Sometimes we still have a really large number of molecules that still look kind of interesting. So we may want to do another medium throughput screen, but maybe it's a small enough number that the PI can do in their lab. So what we do is we reorder the compounds from the supplier to get fresh powder, and then we retest them to make sure they still work, and sometimes they don't. Or it turned out that what we thought was in the well that was given as the structure for this compound actually is not. When you do NMR, you're like, there's a peak of a nitrogen, there's not, things like that. So you always want to double check that everything is what you think it is, and then you start to test the compounds. Usually in the PI's lab, the information goes back to the chemist, and then you start a process of medicinal chemistry to optimize these molecules. So before I go to the chemistry, just briefly, what types of high-throughput screening assays exist, and Bob will probably talk to you a little bit more about next week. But typically, we do either cell-based assays or biochemical assays. There's pros and cons to either, so there's not like, oh, you always need to start with this one, and then the next one is this one. It just depends on the project. So with cell-based assays, the advantage is that it's more physiologically relevant because you're in a cell system and not just an isolated protein floating in a well. So if a compound needs to get maybe to the nucleus of a cell, here you can test whether it actually does that, because otherwise you won't see an effect. And also, some compounds need to be metabolized. Just think about nucleotide analogs. They need to be phosphates need to be added to it. Phosphorylated. Okay. And that can only happen in a cell, because if it's already has a triphosphate on it, it won't be able to get through the membrane. So these kind of things can happen in a cell context. Now, the negative is that maybe a compound would actually be able to get into a cell with a little bit of tweaking, and you're going to miss them here. And it's also expensive. Plus, there's higher variability. It's a more complex system, so inherently it has more variability in it. Whereas with the biochemical assay, the advantage is that if you have maybe your isolated enzyme in a well, you add your compound and it inhibits its activity, you know exactly what it does, right? So it's also cheaper, but you don't know whether your compound will hit other things as well. Yeah, question?
With cell-based assays, do you use primary cells or cell lines?
So with cell-based assay, we typically use cell lines, mostly because we need so many that it's really hard to get enough primary cells of the same batch to run a screen in one go. So sometimes we may have a suboptimal cell line that you do your screening in, but then maybe your secondary screen may be in a primary cell. For example, we have some antiviral assays that we've been running in U19 that we have that are run in Vero cells. But then after you evaluate the activity of your compound in your big screen in Vero cells, they actually go to primary human area epithelial cells to evaluate whether they still work in those. And so it's just a logistic issue that we cannot really use primary cells in a big cell-based screen. But we can use both adherent and non-adherent cells, so that's not an issue. It's just that you need to have enough of one batch to do your assay. So when you look, at one of these charts about drug discovery and development, all of the things in this particular chart that are blue are easy. So, you can have compound libraries, you can buy them, we talked about that. You can identify new targets just every day going to the scientific literature. Actually, the screen itself is not that hard. I mean, at some point you just hit go and the robot does it and it spits out data to you. But then all the other gray areas are bottlenecks. So one is target validation, so that we talked about how do you know if it's a good idea or not. But also assay development, so setting the assay up for the robot to run it correctly. So, and that's because when you go from manually pipetting by a human to the robot doing it for you, the rules change. And what we mean by that is that in a high-throughput screening assay, you need to have a good chance to believe the results of N of 1. Again, you just screen one compound, one time, one concentration. And the reason is it's actually statistically more advantageous to have every compound just one time and cover a broader amount of chemical space. You have a higher chance of picking up a scaffold that will work than if you limit your amount of compounds and test them twice or 3 times. So there's statistics that go into all of that. But so typically the approach is N of 1. But you really need exquisite reproducibility for that. So from well to well, from plate to plate, and also from day to day, because you need to trust the data. So ultimately, you end up with some molecules that look like they do what you want it to. So that's your high throughput screen, your funnel, where from more than 100,000 compounds, you go to maybe a couple of interesting, maybe 10 or so interesting scaffolds or hits, and that then initiates a medicinal chemistry program. So the goal Here's Corinne's lecture, May 4th. So the goal of this program is to establish what we call SAR, or structure-activity relationship. So we will purchase or synthesize analogs, so the kind of brother and sister molecules that have just a slight change. Maybe instead of a 5-membered ring, it's a 6-membered ring, or they add a nitrogen atom instead of a carbon atom, things like that. And you want to establish what parts of the molecule can I mess with to maybe make it more potent, keep it being selective. It needs to have a microsomal stability in that it doesn't get metabolized by the liver really quickly. It's not toxic, things like that. So all those analogs that you buy or make need to be tested for all those parameters. Then the data gets back to the chemist. They get informed and they make their decisions. Okay, this part of the molecule seems absolutely critical for binding to the protein, so we cannot change that anymore. But we really need to improve the solubility. So let's add a hydrophilic group on this other part. That's how their decision and thought process goes. And then ultimately you want to test these compounds for what we call ADME or ADME. I think my next slide is what that stands for: absorption, distribution, metabolism, and excretion. So kind of get a sense of, if this molecule works and you give it to a human, what does the body do to that drug? So the first start is usually, what does the drug going to do to the human? Will it cure my disease, yes or no? But then you need to ask the vice versa question kind of right after that. And then before you do any animal testing for efficacy, we always test the pharmacokinetic parameters in vivo in an animal model, because if you give something to a mouse and it metabolizes it really quickly and 2 minutes later it's gone, you probably don't want to do a 6-month experiment to see if your drug cured your disease, if the drug itself is only there for 2 minutes. So depending on what the application is, your pharmacokinetic parameters, what they need to look like, may be different, but we always want to make sure that it hangs out long enough to have a chance of working. And SAR, there's so many parameters that you need to look at. When you change one thing, it affects all those different parameters, like how does it get metabolized, what's the bioavailability, which means when you take it by mouth, does it get taken up in your gut, into the bloodstream? Is it still soluble or not? What's your patent situation around this? So it's very complex. So somebody compared it once to a Rubik's Cube. You change one thing, you change everything.
So you have these different parameters.
Mm-hmm.
Like which one you could look for first, like target first, like prioritize.
Yes. Yeah, we typically look for potency first, but then we start to look at microsomal stability. So you may actually have a slightly less potent compound, but if the microsomal stability is much longer, then we're going to take that into account. And then as an example for the viral projects that we've been doing, first we just looked at potency, so how little of the compound do you need to give to get, what's your IC50? But then we realized, well, some compounds actually only reduce viral replication by a little bit, and some reduce viral replication by like 10 logs. So you have potency and efficacy, and so driving your IC50 down is one thing, but you also still want to see a really large effect size. And both really need to be taken and balanced in account when you decide what the next compound is you're going to make. So typically we only want to identify one, what we call SAR driving assay. So that is what you're going to look at first to inform the chemist. At some point, especially once you've hit a certain threshold, you really need to start looking at all these other parameters as well. So those ADME studies are alluded to earlier. So what does the body do to the drug? You can do some of that in vitro, so cell culture systems. So for example, does it get taken up in your gut, yes or no? There's a CACO2 cell line that are gut cells of some sort, I forgot what they are, but they grow super tightly together. So if a drug, you give it to the top of a well and then it's a transwell system and you measure how much gets through on the other end, things like that kind of mimic what happens in your gut. And then the microsomal stability I talked about. They're kind of the enzyme factories that we isolate from liver cells. You add your drug to it and you see how fast does it get chewed up. There's a whole enzyme system in your liver that takes care of this enzymatic processes called the cytochrome P450 system. And we can start taking a look which one of those enzymes is mostly responsible for metabolizing. Because there's some that are notorious that metabolize a lot of other drugs. So at some point, when you give it to a human, especially, once you get into older patients, it probably take, a beta blocker, and you start to get polypharmacy. So if humans already take a lot of drugs, you may have influence on those enzyme systems that are going to metabolize your drug. And then in vivo, in an animal model, you can maybe radio-label a compound, inject it, and see where it goes. If you're doing a Parkinson's project, you may want to check it gets in the brain, things like that. And of course, you can try different routes of administration. We typically want to go for drugs that you can take orally by pill, but depending on the indication, you may also do an IV infusion if that's really the only way to get a drug into the system. Because if you're looking at, say a cancer therapeutic area, that may be acceptable. Whereas for treating your toe fungus, you probably don't want to have an injectable. So it depends again on the situation. And Ed Acosta from the pharmacology department, he will give a lecture about PK April 6th. So then ultimately you select your candidate molecule and you want to test that in an animal model for, does it treat my disease, yes or no? But then you also need to test it for toxicity. So you need to do that in relevant animal species, typically in 2 mammalian species, with 1 in rodent and 1 in non-rodent. And you kind of need to mimic the clinical use of the compound. So there's not a formula. You just have to talk with the FDA at some point. What's the most relevant for my disease? Which animal model do you think is the best to go for? Because if, let's say, you want to inhibit an enzyme that exists in humans, also in dogs, but not in mice, you may not want to do your toxicology experiments in mice. You may want to do it in dogs because it's more predictive of the human situation. So a lot of information goes into that choice. And Paul Bush did. He's the director of toxicology at Southern Research. He gives a lecture on April 20th, and it's a really good lecture. He did it last year as well.
So how do you reconcile differences in protein expression and function? So you say it's expressed in dogs, but the protein function might be different than it is in humans, but it's or the expression is different.
Yeah.
So how do you reconcile that with your animal model when you obviously don't have the money to test this in humans, right?
Yeah. So that's why your choice of animal model is critical. So maybe the dog is the best one we have compared to some of the others, and then you talk with the FDA before you do that with those kind of parameters in mind. Like, is this acceptable? Do you think it's a good idea to even test this? And they will give you guidance on that. They won't tell you what to do, but you can go to them with your plan and say, do you agree with this? And then they'll tell you yes or no, and you start a dialogue. Sometimes, especially in mice, of course, we can maybe, using CRISPR-Cas or whatever system, we can maybe include the human gene instead of the mouse gene to make it a little bit more mimicking of the human situation. Many times we do test if a compound's hit a particular receptor, what is the homology, and does it actually inhibit all of the species we want to test it in? And if so, that makes our life a lot easier. If not, well, then we need to have a conversation with the FDA. Does it make sense to test this in this model, yes or no? And also for the efficacy systems. So, for example, in one of our Parkinson's programs, we're actually having a discussion now, should we even do an efficacy experiment in an animal model of Parkinson's? Because we really don't have any good models. So then, yeah, you can do the experiment. What is that going to tell you about the human situation? So there's discussions in the field, for example, with the Michael J. Fox Foundation, where they're saying, actually, let's just look if the compound that we have gets into the brain. Of course, you need to do your toxicity experiments, but then let's just do the human trial, because the animal information for the efficacy is not going to be definitive, so we may as well not do it.
So actually, I work with one of the drugs that you
Oh, great.
So I guess for me, with the LRRK2 inhibitors and the way that they're going, is that the animal models have been difficult just because of the expression differences in the cell that it's in, trying to humanize that model, and the amount of money they're putting into humanizing it. But how hard is it to actually just go from toxicology studies into human trials?
It's not.
Okay.
So unlike common, impression, the FDA actually looks mostly on toxicology studies, and they don't look very carefully about whether your drug cured Parkinson's in a mouse, yes or no. They don't care that much, especially for the first phase. Now, of course, as you, as the investor and inventor, may care a lot because you're going to put in a lot of money in this. So are you yourself convinced that this is worth doing the trial? But the FDA is very lenient because of exactly what you said. The animal models are just not that reflective of the human situation, so they don't require to show that your animal model worked. FDA is much easier than to convince that this is a good idea than, let's say, NIH. So in summary, there's a lot of things to consider. Here's your Rubik's Cube. Your molecule needs to hit your target. Of course, needs to get in your organ of choice. It needs to show efficacy in animals. Again, not by FDA rule, but because if you want to get some money from somebody to get it somewhere, you probably need to convince them. It needs to be not toxic or not toxic enough, depending on the indication you're looking at. What are the pharmacokinetic parameters? And then what's the kind of formulation you may need to do later on. Actually, your phase 1 studies, you can do what we call powder in a bottle approach. You just have, exactly like I said, you just have freeze-dried compound, you add, saline, shake it up, and, give it to a human. Because formulation studies can take a long time, many times this is doing in parallel with your initial clinical studies. And it may change, how much you take up a little bit, but typically not that much to impactfully, kick your program to the curb kind of thing. But it really needs to depend on the indication, what you need to do and how to do it. But at this point, especially studies that need to go to the FDA, they need to be done according to GLP, and it costs a lot of money. So just as an example, we were in one of our programs, we're looking at toxicology right now, like 2-week talks. We want to look at liver pathology, but we're not ready to commit GLP dollars yet. So we did a similar study a couple of months ago, where it would cost about $6,000, like dose a couple of mice, 2 weeks, see how they look. We requested another quote, but we didn't specify we didn't need GLP. Yet a quote came back more than $100,000. So that's the difference we're talking about. So, and it's because all the documentation is needed for GLP, the special training that needs to be done, et cetera, et cetera. So you need to have so that's why we do a lot of non-GLP studies first, because if in the non-GLP studies, all the mice would have keeled over, we probably didn't want to do a GLP study. Good laboratory practice. So there's a lot of documentation, for example, if you need to dissolve something in PBS. What was the lot number of the PBS? When did you buy it from who? Who brought it in? And then when the person dissolved that something in PBS, did they fill out their standard operating procedure protocol and initial there? Who checked that that person initialed? There's so many layers of bureaucracy. It's ridiculous. Well, it's needed to a certain extent, just to make sure that if there's a problem later and we need to go back, and maybe this lot of PBS was contaminated with the fungus, we can recall, or the authorities, just to make sure that things were done appropriately and correctly with the right type of reagents. So the goal then is the first goal is to get to an IND application. So IND stands for Investigational New Drug, and that is for the first time a drug is going to touch a human being. So we have a support office. So Penny Jester actually just retired, but she luckily still answers my emails. So Penny and myself are available for consultation, and I will present a talk about this on May 18th. If you want to know how that works. Lots of paperwork. So then ultimately, and I'll go through this quickly, you have your phase 1, 2, or 3 clinical trials. So like I said, your phase 1 is typically small and healthy volunteers. You give first a little bit, and then you slowly increase the dose. Only in oncology do you actually use patients, and pretty much all the therapeutic areas you use healthy volunteers. And what you really need to look for here is, how does a human metabolize this drug? Does it get absorbed? Do I have any gross effects on organs and tissues, side effects? But these are healthy people, so you can't know yet if your drug's going to cure a disease, yes or no. So that's in phase 2. So it's typically several hundred patients. And here you really look at, does it work? And what kind of short-term side effects do I start seeing? And also maybe what dose range do I need in order to see that effect?
Is that short-term means how long?
Depends on the disease. So phase 1 is typically just, it can be just a one-time administration, or maybe a week or so. But on the phase 2, it really depends on the disease. Is this like a one-time administration, or maybe 10 days if you develop a new antibiotic, or is this a chronic diabetes trial where we need to study at least 6 months, if not longer? So it depends. And in phase 3, this is where you have several thousands of patients, and this is really where you get the benefit versus risk on a population level. And this informs what's on the label. So the little sheet that's in your packet of naproxen that shows, you're going to be nauseous and have rashes and all that kind of stuff, that comes from these trials. So typically you need to have at least 2 phase 3 trials done that show it worked before you go to the FDA and ask the FDA to approve your drug. And Rich Ridley will talk about clinical trials on April 13th.
Two phase 3, that means that the population is different or?
Maybe,
Design,
or
like
just
design.
just independent. No, it should be have fairly similar design. It's just maybe different centers that are involved. It's just replication of, when I repeat the trial, either doing the same time in 2 different places or maybe at the same place, but 2 times in a row, do I get the same results, yes or no? And it depends on, there's so much, it depends. So for some indications, you may actually be fast-tracked and you only need to show a phase 3 clinical trial, or maybe it's really hard to recruit patients because you're looking at people with a genetic mutation of which there are only 1,000 in the whole US, things like that. You may only need to do one.
They usually say, and if that one need to do combine the data from two.
No, they're two. They stay two separate. You don't combine the data. No, yeah. So the FDA will look in the US whether it's approvable, yes or no. That means you can only sell it in the US. If you want to sell your drug in Europe, you need to actually go to European Medicines Agency. If you want to go to Japan, you need to go to theirs. So different parts of the world have their own agencies. There's some coordination now, at least with respect to what kind of forms and what kind of information you need to submit, but they still have their independent judgment call. So then hopefully it gets approved. And then you can start marketing new and improved pill as seen on TV. But will actually continue in safety monitoring. So sometimes this is called phase 4 clinical trials. And especially in a situation where you only had maybe one phase 3 trial done, the FDA may say, okay, we allow you to go sell it, but we want you to continue to monitor. And if there's any untoward offense when we start using this in the population as a whole, we reserve the right to yank it back. And this is also now when you can start repurposing your drug for maybe additional indications. In oncology, you could think, okay, it's approved for breast cancer, and let's look at it in prostate cancer. But sometimes they can be completely unrelated indications. Because of the way the biology works, sometimes one drug can have effects in 2 or more different indications. So that's when you would do this.
So with repurposing, you don't have to go back and show validation. You can go immediately into a clinical trial.
Yeah, so
especially if if it's a completely different disease. I can understand one cancer to another, but in the case of a completely different disease setting, you don't have to go back and show any kind of efficacy.
No, although again, you will probably want to do it yourself before you invest. Sure. But the FDA will, like what we typically do, and we've done several of them on campus with our R&D office. So there was one compound that was used for, what was it? I can't remember what the original indication was, but it was going to be tested in diabetes, which was a completely different indication. And what we did, we got a letter from the company who was selling this drug that gave authorization of the FDA to look into their files with all the toxicology information, manufacturing, all that kind of stuff. And all we submitted was a clinical protocol for this new diabetes trial. So if your population is overlapping in the same, that's especially easy. So this was, adults, otherwise fairly healthy kind of population change. Now, if you go from, diabetes medication in adults to let's treat GBM in kids, it's a bigger step, and you may have to do different safety data in a pediatric population. But other than that, and that's why it's so attractive for especially not-for-profit types of organizations, because you typically cannot make a whole lot of money off of this, because many times those drugs have been on the market long enough, there's no patent protection anymore. You may get a small patent on method of use, so it's not completely worthless, but you can pivot so quickly, and you don't need to spend all those GLP dollars and identify how we're going to make this, what the synthesis route, all the toxicology studies. You don't have to do any of that. You can just quickly do a quick trial, and then hopefully it works.
Have people set up trials without having any patent?
Yep, NIH does it all the time. Yeah. And that may be the proper role for the government when there's no financial incentive for a big pharmaceutical company, for example, for indications that are orphan diseases or only in developed countries. That's really kind of where that's the sweet spot.
So,
Go ahead.
If the drug is from the Japanese companies, then the drug is regulated by the FDA or the Japanese person kind of.
Depending on, so it's regulated a drug is regulated in the place where you want to sell it. So if a Japanese company identified a drug and they want to sell it in Japan, it's the Japanese authorities. If they want to sell it in the US, they have to go to the FDA. So it doesn't matter where it came from. It just depends on where you want to sell it.
Sure. I have a question about patent on this repurposing.
Yeah.
For example, if I develop drugs, first initially I have patent for treating osteoporosis.
Yeah.
But several years later, I find this drug can also be used to treat breast cancer or metastasis. Can I still find a new patent application?
Depends, of course, it depends. I hate that answer. Depends on the claims of the original patent. So your composition of matter patent will typically cover, the physical structure of the molecule. There's a carbon atom and there's a nitrogen atom and there's a fluorine atom, things like that. But many times, patent will also include your method of use, and I want to treat osteoporosis. If you didn't cover breast cancer metastases in that particular patent, you can file a new one. You cannot file around the compound anymore because it's already in the literature. But you could say, and I want to use this drug now to treat for breast cancer metastasis if you didn't cover it
with the new with the new patent date.
Yeah. Yes. And generally, I don't know the details of it. It's considered worth less to have repurposing patents compared to a composition of matter patent, and I don't understand it well enough to really give you a good answer why that is. But I don't know if the patent life is different or what. Sometimes people also do formulation patents. So maybe an existing drug, but they formulate it in polymers that are fancy or whatnot. So now you only have to take something once per month, and you're good for the, that could be a new patent as well on an existing drug already. So just finishing up here fairly quickly, the success rate in the clinic, when you look at, from preclinical to phase 1, phase 2, phase 3, and registration to approval, really the bottleneck tends to be in phase 2. So only 20% of molecules that go into phase 2. And again, this is where you look at does it work or not, make it to phase 3. So this is still the hardest part. So it used to be that a lot of compounds failed in phase 1 where you look at pharmacokinetic properties, but now we have a lot better models in cell culture systems to predict whether a drug is going to behave appropriately in a human, yes or no. So 44% actually make it to phase 1, but it's really, ultimately, we don't understand biology well enough to predict from this outset whether something's going to cure a disease, yes or no. And interestingly, so here, once you go to FDA, 83% of drugs get approved, so it's actually pretty good, but only 1 in 3 ever make their money back, just so you know. So if you're a betting man or woman, don't bet on this. And this is the money slide. So this is where that $1.8 billion comes from. When you look at what does it cost, all these different phases, target to hit to lead optimization, all the way through, launch up your product. And depending on, so they integrated what the chances are of a molecule making it to that phase, depending on data that's out there. When you add up all the cost, including all the molecules that failed, which is a real cost, you're looking at that $1.8 billion. Now, if you are brilliant, and many of us are, of course, here at UAB, and you make the right pick from the start, if you add up all of just what it costs to run all these things, you add it up, you get to $265 million. So even if you were really good at your job, which I'm sure you are, still, this is not something that an entity like UAB can do internally. You need to have a partner at some point and help with this. And of course, this will fluctuate depending on your indication, but it gives you a sense of what this takes. And we only have a few minutes left, so I'm going to skip a few of these things. I did want to point out a few funding opportunities at NIH, especially when you go to NCATS. They do a lot of repurposing RFAs, because that's really kind of that sweet spot that we just talked about of academics and government trying to find a new use for an old drug. The NCI has a program called the Chemical Biology Consortium, and Southern Research is actually a site for those. So that's this kind of similar, like we talked earlier about that screening mechanism. So you can go to the NIH and you say, here, I have this molecule, but it needs to have a lot of medicinal chemistry done on it before it can actually go to clinical trials. The NCI does that in the CBC. And they will outsource then to a location like Southern Research, who does that for different PIs all around the country. So you're not going to get directly the money to do it yourself, but NIH facilitates. They give the money to Southern Research to do it for you. And then other institutes, depending on what your normal institute is you normally go to, they may have either probe development or drug discovery type RFAs, particularly for this purpose. They tend to be specific RFAs because it's kind of an odd duck compared to usual hypothesis-driven research, so be on the lookout for those. And then foundations are actually a really nice resource for drug discovery funding. So we internally have actually a cystic fibrosis foundation grant jointly to Southern Research and UAB for about $8 million. So this is milestone driven. They didn't give us $8 million and say, go forth and do it. It's more like, you go step by step. But we had a trial with them where we screened the FDA approved drug library first. Then they thought, this is going really well. It's easy to work with Southern Research and investigators here at UAB, so Dave Betwell and Steve Rowe. And they said, we kind of want to screen all those million compounds that are in your freezers. And that's where the $8 million contract came from. The ultimately goal is to do chemistry and so on and get to an IND candidate. We have Michael J. Fox. They do drug discovery grants, Alzheimer's Drug Discovery Foundation. There's just a couple. So in your particular field, look at foundations as options because they tend to be patient-driven. So they're really driven many times towards drug discovery and development. So they're a good resource. And then just really briefly, the ADDA, like I talked about, between UAB and Southern Research, we think we can hit all those points in the pipeline from basic research all the way down to clinical trials. And we've kind of fund this valley of death area here, where the basic research is typically funded by NIH. Kind of ends at, okay, now I have an interesting molecule, but a pharmaceutical company is typically not interested until you have at least something that shows it works in an animal model, that's safe, all those kind of things that cost a lot of money. Where do you find the funds for that? So we locally have a investment opportunity to put in some programs. And we have an RFA once per year to solicit for new targets. And then we organize teams around the projects that we fund because this is an interdisciplinary sport. You need to have a chemist on the table, somebody who knows and understands the robot, somebody who understands pharmacology, things like that. So we try to meet at least quarterly. Some of the projects that have been going on for a while, but they're in the chemistry optimization process, we meet at least monthly. Just to kind of keep things on track. And then, this is the last slide where I showed a current pipeline. So we have 4 in CNS diseases. All these red ones here in the middle are in oncology. Then we have 3 antimicrobial projects and then 3 that didn't fit in any of those buckets. Hurler syndrome, kidney disease, and diabetes. So we have 1 that's in phase 1 clinical trials at the NCI. This actually started it was already a compound. We just needed to do a little bit more optimization. And NCI picked it up actually in their CBC program. So they actually run the clinical trial at the clinical center. And then we have a couple that are fairly advanced, like the LRRK2 program, diabetes program, and then we have 1 in multiple myeloma. These 3 are kind of at the point now where we need to invest lots of money because we need to do GLP tox and all that kind of thing. So we're talking with the development office a lot to say, like, we need this much, so many $1 million, because you're talking about 3 million or so each that we would need. So we need to come up with the money somewhere. So with that, next week's lecture is HighThroughput Screening. Bob Bostwick will talk about that. This was really a high-level overview, and because of time constraints, I can only kind of skim over the service. But then hopefully in the next couple of weeks, we can really do a deep dive in all of those different areas, so that you can learn as much as you want. All right, thank you. There's probably still some lunch in the back. And yes, a question.
So with all the failed clinical trials, so they get to phase 3 and then they fail. What steps are you taking, to design better drugs or pick better targets so that you're not investing $400 million for a failed clinical trial and then trying to go back and figure out retrospectively what happened?
I don't think there's an answer for that. So you see, so a lot of pharmaceutical companies are actually getting out of the CNS business. Pfizer closed down their unit, a few months ago for exactly that reason. Because we can do the best we can with the cell culture models and animal models, but if it doesn't translate to a human situation, we still have a problem. So LRRK2 is actually fairly well validated at this point in time, but especially in Alzheimer's, we just don't understand the basic biology well enough. So there's really not an easy answer. All we can do is make the best choices we do with the information we currently have. And then hopefully there's still companies around that want to invest in phase 3 trials in Alzheimer's by the time we, get ready for that. There's no easy fix, unfortunately. Okay.
Thank you.
Yeah, and if you didn't check in when you came in, please do so on the way out with your one card. You just hold it close to the sensor and it goes bloop, and there you go.
Are you going to put your slides in the website?
Yeah, I will put slides on the website, and also I recorded this meeting, so that will go somewhere on the CCTS website.
Thank you.
Mm-hmm.
Great.
I'm going to stop recording.